All right, welcome everybody. Happy Thursday morning. As I'm sure you saw in the share screen, we are going to begin our day with a Kahoot. So let me just, no, that did not work the way I wanted it to. There we go. Go ahead and open up your Kahoot app or go to kahoot.it and put in this code. 3830513 is the number that you need to join in this game. And we'll just get started here in just a minute. We've got 26 people in class today. Let's see if we can get most of you up. As always, as you know, you can put your answers into the chat if you're having any difficulty with the Kahoot. I'll do also my regular check. Can everyone hear me? I assume that you can. Just making sure these headphones are working. Good, thank you. Got a little paranoid the other day when they were not working. If I sound cuts out at any point, please just feel free to unmute and let me know. All right, we're getting close to our goal here. Let's get everybody logged in that can log in. It looks like Justine, your share screen's blurry. I don't know why that is. It could be internet. That's a possibility. Yeah, Marissa, I agree. I think it could be the internet connection. Well, hopefully it improves. As you know, I always call out the questions. Okay, we've got pretty much everybody in. So if you do get kicked out, you know that this number shows up at the bottom of the screen. Okay, Jessica, thanks for letting me know. You can use the chat if you want to, or just listen in. All right, let's do this. Just a few questions to refresh our memories about some things. Shigella has an ID50 of 100. ID50 of Salmonella is 1,000,000. What does this mean? Shigella, ID50 100, ID50 of Salmonella is 1,000,000. All right, so low ID50s, greater risk of infection. Doesn't really talk about the lethality at all. All right, so the yellow people are correct, or I guess that's ochre. Next question, true or false? The most important historical contributors to disease reduction have been antibiotics and vaccines. Is that true or is that false? The most important. I suppose that's open to interpretation, but we're talking about the largest contributor to reducing diseases. It is false. The most important contributions historically have been infrastructure and public health measures. True or false, antibiotics can temporarily worsen symptoms and increase your risk of a secondary infection. Yes, Amber, Annika, that happens to me all the time. It's okay, it's still gonna help you with your learning, so it's a win either way, no worries. That is right, this is true because of if it's a gram-negative infection, we can get a release of endotoxin, and if we are depleting our microbiome, we increase our risk of secondary infection. All right, true or false, an asymptomatic individual can be a reservoir of disease. If you're just joining in, the code to join the Kahoot is at the bottom here, or you can put your answers into the chat. Annika, your mic was not live, so if it was, I would forgive you for that though. All right, that is true. Indeed, it is true. True or false, you can predict the LD50 of a microbe if you know its ID50. All right, that is false. First of all, these are independent metrics. Second of all, the LD50 really refers to toxins, not to the whole microbe. All right, good job, you guys. Shiga toxin has an LD50 of 250 nanograms per kilogram, body weight, if you gave 200 nanograms per kilogram Shiga toxin to 50 people, then what is most likely? All right, good job, you guys. So you would expect somewhere between 0 and 25 people would die. We would not expect everybody to go because it's under the LD50, but this toxin does have some lethality. So we would say something like less than 25. Good work. Exotoxins include all of these except not what? Which of these is not an exotoxin? All right, that would be lipid A. This is the endotoxin, Everybody else is an exotoxin. Koch's postulates were most important because for the first time, scientists could do what? All right, that was the ability to prove causation. Other people had been showing correlations, very helpful. For some time, people had realized that diseases are contagious, and the identification of treatments was coming out for all kinds of reasons, but this was the most direct and important contribution of his postulates, being able to prove, not just correlate, a pathogen with a disease. Koch's postulates may not allow to establish a link between pathogen and disease in which circumstances? That's right. All of these are possibly reasons that would interfere with the postulates as originally written. All right, let's go to the next one. Halfway through, this is a multi-select. Normal microbiota are found where? Select all that apply. Normal microbiota in a healthy person. Where would you expect to find microbes colonizing your body? Yeah, skin and mucous membranes, normal places to find microbes, bloodstream and internal organs, very dangerous and alarming place to find microbes. A hiker's skin knee fails to heal. Instead, they end up with sepsis. This is an example of... All right, welcome back, Anthony. If you need to join in the Kahoot, it's down here, or just put your answers in the chat, whatever is easier for you. All right, focal, right? Starts in one spot, ends up spreading, unfortunately. True or false? Normal microbiota can be opportunistic pathogens. Yes, that is true. Like when E. coli gives you a UTI. Multi-select, which of the following would be symptoms of the flu? All right, subjective things are symptoms that you can describe but can't measure. A fever is you can objectively measure that with a thermometer. If you had said feeling hot, that would be a symptom, but a fever can be measured. So that would be considered a sign instead of a symptom. Sam was exposed to norovirus February 27th, had a stomach ache the 29th, vomiting and diarrhea on the 30th to the 31st. Which of these statements is true? Are these the correct incubation periods? Either one of them. Is this the correct prodromal period? Is this the period of illness? What is the true statement here? All right, yes, the prodrome is the early mild symptoms. If the incubation period was accurate, it would be from our exposure to our prodromal period. So that would be the 27th to the 29th. I know they're quick, aren't they? If 10 people in a class of 100 have a cold, what is the prevalence? All right, that's right, 10%, right? Because we need to express this as total cases out of population as a percentage, 10%. Let's go to the next question. If 10 people in a class of 100 have a cold, what is the incidence? All right, unknown, need more information for the win. We need to know new cases, and this does not break out who has recently caught it or has had it for a while, so it is unknown. What does morbidity tell you? All right, yes, how many people are affected? It's a raw number. The fraction would be prevalence. Severity is unrelated to morbidity, and deaths, of course, would be mortality. Multi-select. New diseases emerge all the time because? All right, all of these things are possible reasons. Excellent. Fomite transmission. Is transmission how? Yes, it's contacting an inanimate object or surface. All right, the parenteral route. Which of these is the best description of the parenteral route as we have defined it in this class, recognizing outside of this class it can have other broader or different definitions. Yes, break in the skin or mucous membrane, such as injection from a needle. Great job, you guys. All right, we got Giardia on the podium. Congratulations to Karen, and also C. diff, of course. Who didn't know C. diff was going to win? Staph aureus, always a good runner-up. Carrie Ann, good job, you guys. And everybody else, I know you were just one step behind, so good work. Okay. Let's go to our lovely slide deck today. Our next job today is to launch into innate immunity, the non-specific defenses of the host. Oops, uh-oh. Hold on one second here. I have craziness happen on my screen. Make sure the screen share is still working. All right, you guys can still see my screen? Awesome. Okay. So the two branches of the immune system, just put them into the chat to get us all warmed up and ready to participate with each other today. I know we've just been participating the heck out of a Kahoot, so you're ready. Yes. The two major branches are the innate and adaptive immune response. Put into the chat any difference you can think of between these two. There are two major ones that I will mention, but just put into the chat any difference that comes to mind when you're comparing these two branches. It could be something very specific, like which exact cells are involved. It could be something general. Okay, great. One is general and one's specific. One is non-specific and one's specific. One it is targeted versus broad. One requires prior exposure, one doesn't. I would amend that to say one gets better with exposure, or one learns from exposures. One has the first and second line of defense, the other is the third line of defense. One defends against any pathogen, the other responds to specific pathogens. One develops through life and one is one you're just born with. Great. Excellent work, you guys. Oh, Lord have mercy. I don't know why this is happening. Hold on. Something is going on with my slide deck. It's just doing something weird. Okay. There we go. Yes, so there's the specificity. The innate is non-specific. The adaptive is specific, although it is important to recognize the innate does recognize patterns. And the innate doesn't change or adapt, whereas the adaptive immune response does. It is also important to note that the two branches collaborate quite a bit. There's several places where we can see them talking to each other and supporting each other. If we think about the last few things that we've learned, we've talked about portals of entry, right? Primarily the skin, mucous membranes, but also the parenteral route. And we care about this because if we're in the immune section now, we are caring about it because this is where we're going to post our defenses, right? We're going to have defenses of all kinds, but we definitely want to make sure we're posting defenses where we're first likely to contact pathogens. We've also talked a lot about virulence factors. What are the tools that the pathogen's using against you? So now that we're in the immune section, we want to talk about how we're going to fight back, right? So let's just see if you can come up with some virulence factors. And it could be any type of microbe. It could be bacteria, virus, whatever. What type of virulence factors do pathogens have that allow them to bind to particular tissues or particular cells? Good, excellent. Yes, so we've got viral spikes, we've got bacterial fimbriae. If you said pili, I would accept that too. We've got membrane proteins and protozoa, which I didn't talk about, but these are major ones, okay? We also talked about virulence factors that allow a pathogen to avoid being phagocytosed. If you can think of anything that might be helpful here, again, you know, primarily we focused on bacteria, but it could be protozoa, it could be whatever. What kinds of things allow pathogens to avoid being found and destroyed by our white blood cells? Oh yeah, great. Yes. A major one is that bacterial capsule, of course, flagella to run away, and especially in protozoans and viruses, I would say, but mostly protozoans, mutation, the ability to rapidly change their surface proteins. Virulence factors also cause damage. Can you think of anything that we've learned about that, you know, and there's been a whole bunch of things we talked about in the pathogenesis section that cause damage. These are things that we're going to be sort of grappling with when we're trying to defend ourselves. Okay, release of toxins, both endo and exotoxins, the ability to explode cells, right? Some of these toxins do that. Some of these viruses do that, damaging membranes, right? Damage to cells. consuming our nutrients. Yes, creating an inhospitable environment due to metabolic wastes. Yeah, stealing iron, exactly, good job. So I have here exo and endotoxins, but you came up with lots of other really good ones. All right, so as was mentioned by another student in the chat earlier, the innate immune response comprises both the first and second line of defense. And the adaptive immune response is the third line that we'll talk about on Tuesday. So you've already done your prep. So what is an analogy that you have for yourself in trying to remember what the first line is for? Okay, thanks, Jessica. Okay, great. And some of you are also just cutting straight to the chase and saying what those components are, and that's also fine. I'll just read through what I'm seeing. So a physical barrier, some of you said the skin, a castle wall or a moat, a protective shell, the bodyguards, I like that one, armor. Yeah, excellent job. So this is mine, but you don't have to use it. I consider the skin to be the wall around the city and our mucous membranes to be like guarded entrances with like a portcullis and a guard. What are the actual components? Some of you have already mentioned skin. So going past the analogy, what are the actual components of the first line? Yes. Oh, I like that guard dog, Marissa, yeah, for the analogy. Okay, yes, skin and mucous membranes for sure. Anything else? Yes, normal microbes. Good work. Okay, now let's do just a quick raise your hand for each of these three so you can tell me if you're feeling comfortable how each of these protects you. So let's start with skin. Who'd like to raise your hand and say, how does skin protect you? Marissa, go ahead.
It prevents things from getting inside your body.
Okay, great. Let's go to mucous membranes. Who'd like to just have a quick word about that? What do mucous membranes have or do that's protective? Karen, go ahead.
They can trap microbes and I think help destroy them.
Yeah, so the mucousy part of the mucus can help to trap the microbes, and then that allows other things that are in here to sort of do their job, whether it's the ciliary escalator, dissolved substances, whatever it might be. And would eyelashes be included in this? That is a really good question. I would say, yeah, I would say so. They are part of your protection, right? They kind of capture things. They protect your eyes. Normal microbes, how do they protect you? Raise your hand if you'd like to say something about that. Karen, go ahead.
One of the things is they just take up space on your body so that other things can't get a hold.
Yeah, they take up space. That's excellent. All right. So here are the things that I would say, and many of you touched on these things. So the skin is a multi-layered dry acidic environment that prevents most microbes from being able to colonize and enter your body. And yes, mucous membranes, they have mucus. Sometimes they have the ciliary escalator. They have dissolved lysozyme, which is antibacterial, and other things. The normal microbiome occupies space and protects that space actively. So it takes up space, uses up nutrients, and actively protects its own environment, either by changing the environment to something that other microbes can't deal with, by acidifying it, for instance, like the vaginal microbiome, or by actively producing toxins that they themselves are immune to but will kill their competitors. So we can see that sort of picture here of our first line of defense, which is the, every one of these surfaces that we have direct contact with the external environment. Some of these surfaces are very good at protecting us, and some are more porous because we do still need to take in light information, and we need to still take in food and sound and so on. The skin, as you've certainly learned many times over, is a multi-layered cell layer that has a low pH, very dry, and the top layers are dead and made mostly of keratin, which is water repellent. It furthermore frequently sloughs, which allows you to shed the surface layer of microbes. Mucous membranes are not as multi-layered, and so they're not as protective, but they do have a columnar shape, and they also have goblet cells producing mucus, and often they'll have cilia so that the cilia can move the mucus against gravity, for instance, in the lungs. So we'll talk a little bit more about the substances that are in mucus and the cilia. We'll look at that as ciliary escalator, moving microbes up out of your bodies, particularly in your respiratory tract. Okay, so you imagine that this is actually sideways, and as the microbes come in through the air because you inhale, they get trapped by the mucus, and the ciliary escalator pushes them up against gravity. What other things do we have? We've got peristalsis in the gut, muscular contractions moving stuff forward. We've got just gravity flow of fluids like urine and vaginal secretions that flow down with gravity out of the genitourinary tract to wash it. So it's like a little waterfall. So if you're going to colonize that, you've got to have virulence factors that allow you to grab on and not be swept away by the stream. And the lacrimal apparatus, which washes your eye. Okay, so let's just take this real quick case study. During a year, nosocomial infections occurred in 74 patients in one hospital. All of these patients were intubated and mechanically ventilated. The infections were caused by something that was transmitted in non-sterile mouthwash, but was not contracted by the patients who used the mouthwash, but were not intubated and mechanically ventilated. Why did this happen, do you think? Just raise your hand if you want to speak briefly to the situation here. Marissa, go ahead.
Were they a pathogen of opportunity?
Certainly, yes. Can you speculate more? If you can't, that's absolutely fine, as to why it was particularly these patients that were affected and not the ones that were ambulatory?
Is it because with them being intubated, they're laying flat and they can't breathe regularly on their own, so it can't be moved out of their lungs, I guess?
Yeah. So being prone is a risk, right? Because it's harder for you to clear infection. Some of these patients perhaps were not, their ciliary escalator was perhaps not working. And we have tubes in there, and the tubes are like little highways that the microbes can crawl along. And possibly, yes, their respiratory system may already have been compromised, putting them at risk of secondary infections too. Okay, so let's talk about something else that's in our first line of defenses, which is not as easy to sort of conceptualize because it's stuff that's dissolved in our secretions. In our sebum, which is secreted on the skin, there's a fungistatic fatty acid. Our skin is also quite salty because of sweat. What genus is famously able to survive high salt? Do you remember? Put that in the chat if you remember. It's a genus that's common, many species of which are inhabitants of the skin because they're able to survive here. Yes, Letitia's got it. Yeah, that's right, you guys. Members of the Staphylococcus genus are salt tolerant up to 10%. And this in part explains why they're more likely to be involved with skin infections. Now, the low pH of the skin is achieved on the skin, as well as there's low pH in your stomach, of course, partly in order to unfold proteins for digestion, but also partly to kill microbes. And there's a low pH in the vaginal secretions as well that's established largely by normal microbiome. And all of these are helpful, but why? Why is it that obviously things can live in this low pH environment, but why is low pH helpful? Okay, it denatures proteins, Alex, that's true. Do you remember when we talked about sort of where most microbes lie on the spectrum of their pH preferences? Yes, most pathogens, not all, but most of them are neutrophiles. So this is a very useful way of kind of keeping down the microbe load on your skin, in your stomach, and in the vagina, though you will still have normal microbes that live there that can survive these conditions. Now, lysozyme is also in your secretions, pretty much in all of your bodily fluids and your secretions. And what does it specifically target? As a hint, bacteriophage use it too. So it's a common tool that we share, which is really interesting. It's found in our sweat, tears, saliva, and urine. My favorite thing to say is if you cry, you're protecting yourself. And so, yes, it breaks down cell walls. Specifically, it chops between the two sugars of the peptidoglycan filament between the NAG and the NAM. So its action is somewhat similar, but actually molecularly distinct from penicillin, which also attacks peptidoglycan, but in a different way. And so it's just it's a different enzyme, but it does target bacterial cell walls. Last but not least, we have transferrin, and this is in your saliva and other places too. Does anyone know what transferrin does? Yes, that's right. Exactly. It is iron binding, trying to keep that iron away from bacteria. Good. All right, so we've already covered what lysozyme does, which hydrolyzes peptidoglycan, and the link between the NAG and the NAM sugars. And I just love the fact that we share this defense mechanism with phage that use it as a tool to get into and use their host bacteria. And it helps us by being generally antibacterial. It's found in all our bodily fluids. And then transferrin, as many of you mentioned, binds iron. This helps you because A, it protects you from iron toxicity. And make sure you have enough iron, but importantly it prevents bacteria from getting access to iron, and is important especially if you are infected with a bacterium that is actively trying to break your blood cells open. So you need something to go and sweep up that iron. Okay, let's hit the final part of the first line of defense, the normal microbes. They competitively exclude pathogens by covering up the space of binding sites, right? They're sitting there in the way of those cell receptors that would normally be used by viruses, let's say, or in the way of the parts of the cell that could be bound by fimbriae. They eat up the food so the pathogens can't get there. They alter the environment. Can you think of any metabolic process that, for example, changes the pH of the environment? When we think about metabolism, is there a form of metabolism that routinely creates a different pH? Yes, it's through the waste product of this metabolic process. Think about the major metabolic pathways. There we go. Annika's got it. And using your question marks. Excellent job, you guys, using your question marks. I like it when you use 2 of them. That's very helpful. Yeah. Fermentation often produces acid byproducts, which changes the environment. So that's how some of those microbes, for instance, in the vaginal environment are able to achieve a low pH environment. There are other metabolic processes that might change the pH in a different way too. And some will actually make antimicrobial toxins, something that they have some immunity to, but are toxic to other microbes. Now, commensal microbes are part of our normal microbiome. Put your best definition of commensal into the chat. Thank you, Alex. I love that. You provided an answer key. 1 is not sure, 2 is like, I'm kind of lost. I love that. That's fantastic. Very helpful. Okay, yes. One party benefits, the other doesn't care or is not harmed. Good job. Yeah. 1 benefits and 1 is unharmed. Okay, so in a relationship, it's all symbiosis means it's a relationship. So that relationship could be great, medium, or bad, right? But in all of those relationships in the microbial world, at least one of the organisms is benefiting. The question is, what's happening with the other guy? In a commensal relationship, one is benefiting and the other is neutral, not harmed, not helped. In a mutualistic relationship, both are helping each other, and in a parasitic relationship, one is getting a benefit at the expense of the other, which is being actively damaged. Good. And so commensal microbes in our normal microbiome are the most likely ones to be opportunistic pathogens, in that in one setting, they're just taking up space and not really doing us any harm. But if they get into the wrong part of the body, they may actually actively harm us. And so UTIs are a good example of that. But so are skin infections, right? Things on the skin like staphylococcus may be helpful there or neutral there. But if you have a break in the skin, they can cause an infection. All right, and as we've talked about at length, the effect of antibiotics on normal microbiome is to disrupt that normal balance, and this can allow pathogens or opportunist pathogens an opportunity they wouldn't have had before. Okay, so that's the first line. Let's talk about the second line. We've got phagocytes, we've got inflammation, fever, and antimicrobial substances, more substances. All right, so what are the goals of the second line? This is where it gets interesting because this isn't just like a sort of passive physical barrier. There is actually some kind of recognition. So the analogy here would be more like beat cops patrolling an area, not with any particular agenda, but just kind of looking for trouble. And if you would prefer to come up with a different analogy because that one doesn't feel very kind or good to you right now, then feel free to do that. But it's still more active. You know, it's sort of, there isn't a lot of specificity. It's not like a detective that's on a mission and on a case, but nonetheless, they're looking for trouble. They're looking for a pattern that says safe versus danger or us versus them. And the pattern recognition is primarily of bacterial signatures. So stuff that's found on bacteria, that's found on lots of different bacteria, that's kind of says there's a bacterium there. We don't know who it is. We don't really care. We don't know the details. We're not going to be specifically aiming for this guy, but there is a bacteria there. So raise the alarm. And so things like bacterial signatures, certain kinds of molecules on the surface of bacteria, or molecules released by tissues that are being damaged. Okay, now if we look at all the stuff, the cells that are in your blood, right? We've got red blood cells, we've got platelets that are not cells anymore, but were derived from the hematopoietic stem cells. We've got the white blood cells, we've got macrophages, dendritic cells, neutrophils, T cells, and B cells. So some of our blood cells are not really involved in immunity at all, right? Red blood cells and platelets are involved in oxygen transfer and preventing you from bleeding out. And some are involved in the adaptive immune response, T's and B's, which we'll talk about next time. But all of these guys are important in the innate immune response. And in particular, we've got these 3 phagocytes whose main job is to go and gobble stuff up. And so there's a couple of ways of analogizing these. Macrophages, I think of sort of like as deployed troops. Dendritic cells are more like scouts, kind of feeling the environment out. And then a neutrophil would be maybe more like a first responder. So let's have a look at those in a little bit. One thing I want to mention is, some of you may already have heard about this, that there's sort of a crude way to kind of get a guesstimate of what's going on inside of somebody by just doing a simple white blood cell count. So by taking a blood smear and then by looking at which types of white blood cells are in there in what ratios, and this can be done under a light microscope because these different types of white blood cells have different appearances, and it takes a little bit of training, but then you can get there pretty easily, and you count your first 100 white blood cells and you assign them to one of these categories and see if the ratios are normal or abnormal. So these are the normal ratios, okay? Neutrophils between 2/3 to 3/4. Basophils and eosinophils usually pretty rare. Monocytes, around 5%, and then lymphocytes between 1/5 and 1/4. This would be normal in a healthy person. If we see an unusually high level of neutrophils, what could this mean? Yes. Good job. It's usually an early sign of infection because these are going to be the first to be deployed in an increase in number during an infection. So if we see an increased number of neutrophils, it's likely to be early in an infection. An increase in basophils, what might that be associated with? Basophils secrete histamines, and so they're commonly associated with inflammation. And sometimes when our immune system is sort of getting aggravated by stuff that's not actually dangerous, but it just thinks it is, it's involved in allergies. All right, eosinophils. A high load of eosinophils, what could that tell us? And they're normally like around 3%. So we see larger numbers. What might be going on? Yeah, good job, you guys. Exactly, a parasitic infection. These are primarily involved in fighting parasites. Although, again, this part of your immune system can be sort of misguided and help to generate allergic responses. Monocytes. Monocytes are a precursor to macrophages. An increase in these mean, well, it's a little bit more general. Yeah. It typically means some kind of infection, and it might mean that there's a chronic infection, right? That we're sort of later in the stage and we're not clearing it. Lymphocytes are B's and T's. And so if we see more of these, then what we can reasonably guess is that our adaptive immune response has kicked in. Of course, if we see a decrease, especially in our T cells, then that could mean is that our immune system is actually being damaged. Okay, great. Okay, so I'm going to show you this film if this works. Let's find out. Let's see if my screen share is still optimized for sound. It does seem to be. All right, so we're going to watch this together. Let's see if it works. Dang it, it's not working. Oh dear. Okay, hold on one second. I have to enable the content, and I think now it's going to work. All right, now I have to screen share because I don't think you can see this right now. All right, let's find out if this very fun little video is going to work for us. Yes. Okay, I've got some questions. Let's start here. All right, first question, sort of basic question is, this is an image, this is a light microscope with a time lapse. So we're imaging it with a light microscope. So that's just some context for you. What are these blobs here? Just write into the chat what you think the sort of like dozen blobs on the screen here. You can do it, yes. Thanks for encouraging it. Okay. Most of you are guessing on a cell that's a little bit too big. These blobs here are not the white blood cells, but yes, some of you got them. There we go. These are the red blood cells. These tend to be about 10 micrometers wide. And then what's this guy here? Yeah, a white blood cell. They tend to be about 30. They're bigger, right? 30 micrometers or 20 or something like that. And it looks to be specifically not just any white blood cell, but a phagocyte. So it could be macrophage, a neutrophil, or dendritic. But in the beginning of the video, it actually said it was a neutrophil. Now he's chasing something. Based on the information that I've given you so far, what do you think he's chasing? And just from looking at the video, like a bad guy. Okay. Yes, a motile bacterium. Yes. So some of you have also guessed parasite or toxin. And so let's just address each of those. We can actually rule them out on the basis of just what we're seeing. This is a light microscope. So a toxin is one protein. So that's going to be way below the level of resolution. It's also not going to be a virus for two reasons. It's not a virus because, again, that's too small to be seen with this technology. And viruses are not motile. As some of you mentioned, this is motile. It's running away. And so it's probably got flagella on it, right? And it's probably sensing that it's being chased and it's running away. Okay. Let's see what else you've said. Yeah, bacterium. How do we know it's not a parasite? Parasites are usually eukaryotic. How do we know it's not a parasite? That's right. It's too small. It's the perfect size to be a bacterium though, right? If this is 10, right? This is like maybe, I don't know, 8 times smaller than that or something. So it's the right size to be a parasite, or a bacterium, sorry. All right. So let's talk about the phagocytes. That was a neutrophil, but there's several types. The word phagocyte means, it literally comes from the Greek to eat cells. And it's an ingestion process where you grab something, and the something you grab could be much smaller than what we were able to see in that video. Could be a toxin, could be a virus, possible, all possible. I talked about how the neutrophils are considered to be like first responders, and we'll see a little bit more about that in a second, because they're usually sort of very short-lived, and they're produced at high levels early in infection to kind of like run to the scene and see if they can take care of business right away. Dendritic cells are circulating all the time, kind of scouting for information. So when they're deployed, or they're kind of always deployed, but their job is not to be primarily responsible for clearing the infection, even though they will ingest things, but primarily it's for gathering information to talk to the adaptive immune response. And look, there's one of many examples that we'll see where that innate immune response goes and talks to the adaptive immune response. And macrophages are like deployed troops or a SWAT team. They're very damaging and dangerous. So typically they're put out in the body in an unactive state, and they have to be sort of switched on, because you don't want them rampaging through the body if there's nothing going on. Okay, so I would like you to list the steps in phagocytosis on a piece of paper, perhaps. And then we'll just kind of check in a second. I'm going to sort of say what to do with that in a moment. But first, just take a piece of paper and just kind of jot out the steps in phagocytosis. Yes, Annika. Dendritic cells wandering around yelling, "Look what I found. Hey guys, look what I found." Exactly. Yes, very good analogy. So make sure you have like your steps kind of itemized, just like 4 or 5, whatever you think the steps are in phagocytosis. I will do just a smidgen of cold calling to kind of keep everybody awake. And as always, you know, the rules of my cold call is don't be afraid to speculate, but also don't be afraid to say, I don't know, or I want to pass. That's completely fine. Okay, so let me see who I'm going to bug first. Michaela, what would you say is the very first step in phagocytosis? Are you able to unmute, Michaela? I think there might be a microphone issue there. How about Letitia? What would you say? Oh, I can hear you now. Go ahead.
Can you hear me?
I can hear you. Go ahead. Oh, it looks like we're still having microphone issues. Letitia, was that you?
Yes.
Okay, let's see. Let's go to Paisley. What would you say was the very first step?
I have to find and bind.
Yes,
So
we got to find and bind. We got to chase them down and grab them. Excellent. Yeah, Michaela, I'm sorry your mic isn't working. That's what I figured. Okay, let's go to Alex. Once you find and bind, what's the next step, Alex?
Ingestion.
Yeah, we got to endocytose them, right? We got to bring them inside. Yes. Christine, after we bring them inside, what is the next thing that has to happen?
Once they're inside, breaking them down and pulling them apart.
Yeah, we've got to digest them. We first ingest them, then we got to digest them. Okay, great. So good job, you guys. So let's look at this kind of in a little picture here. So we've got the first step, which is we've got to grab onto them. Okay. And there's some molecular details here that we won't go into in great detail, but which if you take this again in a subsequent course, you'll be challenged with things like the toll-like receptor and PAMPs and all kinds of fun things. So there's more molecular details here that I'm really kind of revealing. So first we got to grab them. Then we've got to endocytose them using our little extruded membrane called pseudopods. But this is an endocytosis known as phagocytosis because we're taking in a whole cell. And that creates something called a phagosome, Okay, you do want to remember that term. Now we want to digest them now, and so we've got to take our lysosome and fuse it to the phagosome, and this creates something called a phagolysosome, okay? Next, these enzymes in the lysosome go to work digesting this all up, and then we spit out the pieces, okay? All right, let me have a look at our time. We are actually right on time for our break. So let's take a 9-minute break just so I don't run out of time. And let's reconvene at the top of the hour at 11 is when I will see you again. All right, guys, welcome back from the break. So let's keep going. Now, phagocytosis, this process we just talked about, isn't always successful. So what is it that some bacteria use as a defense? What is their defense? How do they fight back against us? Just put something into the chat if you can think of anything, any tools they might have to avoid this process. We talked about one of them earlier. You're welcome to use that one again. Great, excellent, lots of good answers. Yes, fantastic. Even some that I hadn't put in my own slides, but good job. Okay, so we've got some of the virulence factors like M protein that prevent phagocytosis, or capsules that prevent phagocytosis in the first place. They prevent that first step, the finding and binding. Some that are more aggressive prevent the first step by killing the phagocyte, like I shoot you first. And then also, running away, so flagella and pili, mutation in their antigens. Yeah, so some of the phagocytosis is happening in concert with the adaptive immune system. So things that are coated with antibodies, as we'll learn on Tuesday, make them attractive targets for phagocytes. So if you change those, you're going to have less likelihood of being coated, so the phagocytes won't be as easily able to find you. Escaping from within the phagocyte. And yes, okay, so here are just some examples. You don't have to remember these species, but just these techniques. So some of them inhibit adherence using M protein or capsules. Some kill the phagocytes with leukocyte and toxins, or lyse them using the membrane attack complex, using pores that will break them open. Some get into the phagosome, that first endocytic vesicle, but they escape from it. And that includes Shigella and Rickettsia. These guys use that comet tail that we talked about. So they use a comet tail and they secrete a substance that causes actin subunits to assemble behind them so they can bust out of cells and out of parts of the cell. Some of them prevent the phagosome from fusing to the lysosome. That's what Mycobacterium tuberculosis does. And some can actually survive in the phagolysosome. An example is this guy, but again, you don't have to memorize these species. Okay, so I'm going to watch another video. If this works, let's find out. I don't know if it's going to work. We're going to look at neutrophil migration, and it looks like it is opening up, but you can't see it, so hold on.
The white blood cells are an important...
Let me just share my screen so that you can see it. All right, let's watch this just for a moment.
The white blood cells are an important part of our body's immune system. Neutrophils are a special group of white blood cells that play an extremely important role in protecting the body against infections. Mature neutrophils do not divide. New neutrophils are constantly formed in the bone marrow where they develop and mature. Mature neutrophils are released into the blood where they circulate for 3 to 12 hours and then move to other tissues where they survive only 2 to 3 days. These mature neutrophils can be found in large and small blood vessels, as well as in the tiny capillaries in different tissues. In the bloodstream, neutrophils act as surveillance cells searching for infections. Neutrophils can sense a site of infection because of chemicals that are released by the bacteria. Upon detecting these chemicals, neutrophils slow down and begin to stick to the walls of the blood vessel. They then squeeze between the cells of the blood vessel wall and enter the infected tissue. Here, the neutrophils crawl towards the infection as they continue following the chemical signals. At the site of infection, bacteria are coated with antibodies of the immune system and marked for ingestion. The neutrophil recognizes the marked bacterium, engulfs it into its body, and traps the bacterium in a sac called a phagosome. The neutrophil contains pouches called lysosomes, which are full of digestive enzymes and chemicals that can kill bacteria. The pouches, or lysosomes, then combine with the phagosomes containing the bacteria and release the enzymes and chemicals, resulting in the ultimate death of the bacterium inside the neutrophil. After killing and digesting the bacteria, the neutrophil is spent. Having completed its duty, the neutrophil shrinks in size and breaks up into smaller pieces that can be recognized and eaten by other cells.
Okay, so that video nicely illustrates a few things that are more jargony that we'll talk about. The steps in migration. So first, the white blood cells are, in particular the neutrophils in that video, circulate in the blood, and they receive signals from tissues. Endothelial does, the white blood cells themselves do, that when the white blood cells receive those signals, it causes them to slow down, and then they get close to the edges, which is those endothelial cells lining the blood vessel. And together the cells grab onto each other, which allows them to start rolling along the inside. This process is called margination. Then they use chemotaxis to follow the chemical signals. And then they do this process you saw before, which I think is just amazing. When I learned about this, I was like blown away. Diapedesis, which is a fun word that just means they squeeze in between the blood cell wall cells. They squeeze out of the vessel, but they're doing it because the vessel is not just a tube like a straw. It's made of cells, so there's little spaces between those cells. And then they follow more chemical signals to find their targets, and often their targets are coated with antibodies, which of course is another example of how the innate and adaptive immune response talk to each other. Okay, so that's phagocytosis. We're going to talk about inflammation next briefly and fever, and then we're going to spend a lot of time talking about one of the particular groups of proteins that protects you. Okay, so inflammation is just this process of you get some kind of wound. It could be external or internal that causes damage to the tissues and potentially can also introduce bacteria into places where it shouldn't be. So this could be due to a puncture wound, or it could be due to a trauma like internal damage during, due to impact in a car accident or a sports injury. And this causes increased blood flow to the area that brings neutrophils, monocytes, and lymphocytes to the area so that we can get our first responders and our deployed troops and our adaptive detectives out on the case. We get clotting to prevent you from bleeding out and hopefully to stop the microbes from spreading. And then eventually, as any microbes are cleared, we get kind of a debris accumulated of dead neutrophils that have grabbed on the things that they're trying to get rid of. And this is what actually forms pus. And then as a result of that, you can either have acute inflammation, which is sort of the ideal because it starts and then it ends, or chronic inflammation, which is not ideal and can lead to long-term damage. Justin says that about diapedesis, I see the vessel as a subway and then getting off at their stop when they squeeze out. That's excellent. I love that. Okay. So if we think about inflammation, one of the hallmarks is vasodilation. Why do you think that this is helpful? Why would you want your blood vessels to enlarge if you're in the middle of an inflammatory response? Just put into the chat any ideas you have about that. Okay. Yes, that increases blood flow to the area. Is there anything else? Like we got, you know, we're bringing a lot of fluid to the area. Can we say more about like, why does increasing the size of the blood vessel, which would make the distance between cells in the blood vessel a bit bigger, what might that facilitate? Yeah, okay, wouldn't it make the white blood cells have an easier time doing diapedesis, getting out of the blood vessel and into the tissue? Okay, so we're doing vasodilation increases blood flow, so we get more guys coming to the area, but then we also get an increase in the gaps between the cells of the blood vessel, which makes it easier for the white blood cells to exit the blood vessel and enter the tissue. So what are the hallmark effects of inflammation? Redness, yes. Heat, swelling, and pain, right? Yes, all of those are just when we say it's inflamed, this is what we're talking about. And the swelling from extra fluid, right, in the tissue, pain because there are pain receptors that are being activated, heat because of what's all is happening there, and the redness as well. So that's inflammation. Now, a fever is a different kind of heat, isn't it? It's an overall systemic increase in body temperature. And so it does increase our body temperature. We can talk about the disadvantages. Some of them are very serious. What would be the advantage of a fever? Why would increasing our body temperature be a useful thing to do? Yes, good. Okay, excellent. I sort of see three related answers in the chat. So one is the idea is that hopefully this will kill bacteria or viruses. And some of you have gone deeper into the molecular reason, which I love, which is to say that the proteins are hopefully getting denatured. And also some of you have talked about how, you know, if a bacterium is adapted to a particular heat environment, raising the temperature might make it might inhibit its ability. But again, that comes down to the fact that we're denaturing the proteins and potentially liquefying their membranes. A few other things that maybe are not as obvious is that it increases the ability of transference to bind to iron. And maybe that doesn't seem like a big deal, but remember that bacteria have their own iron binding agents. What is the name of that special substance that bacteria use to bind iron? This is something you do need to know. This is a word that you want to put on your list. There we go. Carrie Ann's got it. Siderophores, exactly. Now, in a tug of war between siderophores and transferins, on a regular day, who's going to win? Who's stronger at binding iron, siderophores or transferrin? Yes, that's right. Bacteria are better than you again. Siderophores are stronger. So a fever kind of boosts our transferrin's ability to kind of tug back on that iron. It also increases the ability of some of the cytokines, these chemical signals that white blood cells use to talk to each other and to talk to tissues in your body. So it increases their ability to be sort of sent and received, and it produces interferon, which we'll talk about shortly. And it does also, hopefully, slightly denature maybe viral spikes or maybe fimbriae proteins and so on. However, of course, you are also made of cells that rely on proteins. So there's the disadvantage there that high fever can also, in fact, affect your proteins and membranes. What would be sort of the systemic consequences of that? What are the disadvantages of fever, looking at it from the perspective of sort of symptomology or signs that are showing up in the body? Yes. So it can affect our heart rate, and it can affect our hydration level and the pH. A very high fever is fatal. So we don't, you know, it's one of those things, it's kind of like a chemotherapy, though of course it isn't, which is you're trying to hit that sweet spot between damaging your opponent before you end up killing yourself. All right, so let's focus the rest of our time on antimicrobial substances. And a lot of that is going to be on the complement system. We will have 2 shakes of a rabbit's tail talking about interferon towards the end and revisit some of those other tools that are considered antimicrobial substances devolved in your bodily fluids. But here's the complement system, and this is a very useful little graphic that kind of indicates that there's like, I like to think of the complement system like a light switch that can be turned on. And when you turn it on, boom, a cascade of events happens like dominoes. And so you get 3 outcomes. And the way to turn on this light switch, there's 3 possible ways to do it. So it's named the complement because why? Does anyone know why it's the complement? And this is complement, which means to help, not compliment, which means to say nice things about. What is the complement system complementing? Yes, it is helping the adaptive immune response. So it is definitely part of the innate immune response. Why? You have it as soon as you're born. It doesn't change. It's more relies on pattern recognition, but there is one place where it does talk to the adaptive immune response and helps it. So just like phagocytosis, it can be activated by the adaptive immune response. All right, so the complement system consists of 9 proteins. What are they called? Very creative name, super hard to remember. Yes. C for complement, C1 through C9. And they're found in all your body tissues, circulating in blood and bathing your tissues, the body fluids that are bathing your tissues. And typically as they're circulating in their body, are they active or inactive? That's right. So they circulate in an inactive form and they can be activated. So how they get activated, there are 3 possible ways to activate them. And then this light switch actually is a sort of complex mechanism consisting of several different pieces that kind of get triggered one after another. But though there is one guy kind of at the top of the cascade chain. Yes, Annika, we used to get cool names and now we just get numbers, it's true. So the central cascade is sort of the mechanics of the light switch. Like the first thing you flip on is that little light switch, but then there's like wires and stuff inside that as soon as you flip it on, these things happen and they happen really fast, but they're all part of the mechanics of it. We'll talk about that. And that ultimately lead to these 3 outcomes. So the central cascade consists of C3, which gets broken into 2 pieces, C5, which gets broken into 2 pieces, and C6, 7, 8, and 9. There are 3 pathways that activate this light switch. What are the 3 pathways? Yes. That's right. The classical pathway, this is the one that talks to the adaptive immune response, hence the name. The alternative pathway, because it was found second and they were like, oh shoot, there's another way. And then the lectin pathway, which thankfully is named after the protein lectin, which is part of the activation mechanism. And last but not least, once any one of these 3 pathways has been activated and the light switch gets turned on and all the dominoes fall, what are the actual outcomes that we get? Yep. Great. So it triggers an inflammatory response. It also triggers cell lysis, so attacking foreign cells and basically exploding them. And it triggers an opsonization event where we coat targets with chemicals that make it easier for phagocytes to find them. So let's begin by talking about the mechanics of the light switch. Okay, so here we've got all the different pieces, right? We've got C3 at the top of the heap. When that's activated by any of the 3 different pathways we talked about earlier, it gets activated, which means it's broken into 2 pieces. So in its inactive form, it's whole, and when you break it, it's now active. One of those pieces called C3A goes on to participate in inflammation, and the other piece has 2 jobs. It's part of opsonization. It coats the surface of things that we want to get rid of with phagocytes. and it goes on to activate C5. And by activate, I mean break that in half. C5 breaks into two pieces. The C5a piece goes on to participate in inflammation along with C3a. And C5b has the very important task of recruiting the rest of the C proteins to form a hole that sits in membranes and explodes cells. All right, so that's the sort of mechanics of the whole thing. And so as soon as you have activated C3 by breaking it into two pieces, then the rest of this just happens like one thing after another, like a domino effect. Let's talk about how we activate C3 though, because there's three methods, and there's the classical method first. So the classical method uses the first C protein called C1. What you can see is it requires that the adaptive immune response already has generated antibodies against something, and those antibodies bind to their targets. The stem of the antibody, which is not the binding part, is a trigger for C1. So C1 finds those little stems stuck to things and it says, oh, I need to get activated. And so C1 then talks to C2 and C4 and activates them by breaking them in half. Two of the pieces, one from each of these guys, create something called C3 convertase, which then breaks C3 in half. And you don't have to remember which of these pieces, just know that C2 and C4 are involved. So for the classical pathway, C1, C2, and C4 are required to, and they get a signal from an antibody, and that C1, C2, C4 leads to C3 activation. And then once you've gotten C3 activated, boom, all the things happen: inflammation, opsonization, and cytolysis. And this is a really important place where the adaptive immune response talks to the innate immune response, because the adaptive immune response is all about generating antibodies. But then once those antibodies have found their targets, all kinds of aspects of the innate immune response can kick in. They don't need to know about this part here. They just know an antibody has found its target and it's like their turn to shine. Okay, now we talked about C1, C2, and C4 being required for the classical pathway. What proteins are important at the beginning of the alternative pathway? How do we get to C3 via the alternative pathway? Yes. The alternative pathway does not rely on the classical pathway relies on antibodies. So this circumvents that altogether. This is completely innate. There's nothing about adaptive immune response required here. Instead, there are these three proteins called B, D, and P that are able to bind to sort of patterns of lipids and carbohydrates that are found on bacteria only. So these are recognizing patterns that are commonly found on bacteria and not found on human cells. Paisley, go ahead.
If those are the proteins, then what's the C3A and the C5A?
So C3A is half of the C3 protein. That's part of the central cascade. So when you break C3 in half, you get C3A and C3B. And then C3B goes on to break C5 in half, and that creates C5A and C5B. And we'll sort of revisit that again, Paisley. And then if at that point it's still unclear, please do ask me to go through it again. Okay, so what B, D, and P do that's special is they don't require the adaptive immune response at all. They already come into the world knowing that there's some patterns that are found on bacteria. And they find those patterns, they bind them. They're like, hey, there's a bacteria. We don't know if it's E. coli or Shigella or Staphylococcus aureus. We don't really care. This is a bacterium. It's dangerous. And that recruits and activates C3 directly, bypassing C1 and C2 and C4 altogether. And then what happens next is we've now turned on the light switch. So as soon as this binding event happens between BDP, its targets, and C3, then C3 cuts itself in half and we get these two pieces, C3A and C3B, and then the whole cascade starts. This goes on to participate in inflammation, this participates in opsonization and activates downstream events, including breaking C5 in half and creating C5A, which goes on to inflammation, and C5B, which goes on to cytolysis. And we'll take a closer look at those downstream events in a second. Yes, it is, Lynn, usually. So it's particular signatures of lipids and carbohydrates together on the surface of the microbe on the cell wall. Okay. Now, the third pathway, called the lectin pathway, named after the protein lectin. Let's first ask you, what part of your body makes lectins? Yes. Correct. It is the liver. I like to tell my nutrition students that if they're ever in doubt and they're not really sure what the answer is to a question, guessing the liver did it is a good guess. Okay, so we have lectins made by the liver. They don't need C1. That's something the classical pathway needs to recognize antibodies. So again, there's no antibody component. This is again bypassing the adaptive immune response. It's innate all the way. Lectins are made by the liver, and they are capable of recognizing another pattern that's found on bacteria. And that is carbohydrates containing the sugar mannose, which is not really a human pattern. So it recognized patterns on bacteria because of the presence of the sugar mannose in complex carbohydrates, again on the surface. This recruits and activates C2 and C4, just the way that C1 did. So it's sort of taking the place of C1. And breaking this in half creates C3 convertase. And that activates C3 by breaking it in half. And then the two pieces of C3 go on to participate in inflammation and opsonization respectively, and activate C5 for cytolysis. Okay, so the liver makes this lectin that binds to mannose. This activates C2 and C4, which activates C3, and then we get opsonization, cytolysis, and inflammation. So now let's take like a sort of go down the pathway a little bit and look more specifically at these three outcomes of complement activation. So inflammation involves which parts of the complement, which C proteins. Yes, lectin's just floating around waiting to see if it binds. That's right, Alex. Which parts of the complement are directly involved in inflammation? Yes. C3A and C5A. And if this helps you at all, C3A for inflammation and C5A for inflammation. Okay, that's how I remember it. So if that helps you, great. If not, try to rinse your brain and ignore what I just said. Opsonization most directly involves what? What part of that central cascade is important for opsonization? Yes. That's the other half of C3 called C3b for like, I think of it as like binding because it coats the surface of things that we want to get rid of with phagocytes. C3b is involved in opsonization. What do we need for cytolysis? That's right. We need C5b, so the other half of C5 that's produced when you break C5 in half. The C5a, of course, being involved in inflammation. C5b is involved in cytolysis. How does that work? It recruits C6, 7, 8, and 9 to make a pore, and this is the outcome of that. You can see what happens when a cell is targeted with this cytolysis. It goes from a nice intact cell to a very not happy cell. All right, so those are the proteins that are required. A little more info on how C3A and C5A actually work to promote inflammation. They bind to receptors on the surface of histamine-releasing mast cells, and the binding of them to the surface receptor triggers the release of these little vesicles containing histamines, and histamines are pro-inflammatory cytokine. Also, C5A itself is a chemoattractant for white blood cells that are phagocytes, for neutrophils and macrophages. If this gets released, then neutrophils and macrophages get really interested and they come in from the bloodstream to the tissue. Okay, so C3A and C5A bind mast cells. That causes them to release histamines, and that increases the permeability of blood vessels. And C5A is also a chemoattractant, so that as these guys are floating through and they receive the signal, the blood vessels already been enlarged to make it easier for them to follow the rest of the trail. Opsonization, that was achieved by C3b. is important to help coat the surface of microbes that we're trying to get rid of, or whatever it might be that we're trying to get rid of. And so it makes it easier for phagocytes to find. It's like a process of coating these things so the phagocytes know what to eat. All right, and then finally cytolysis, which requires C5b to bring C6, 7, 8, and 9 together to form a pore that pokes holes in membranes and leads to their cell lysis. All right, so let's see if we understood that well enough to answer this application question. A patient with a genetic disorder that affects one of the complement proteins would have the most severe disease if which of the complement proteins was the affected one? Which of these mutations would cause the most serious disease or defective complement? Great. Yes, it's C3. Raise your hand if you want to explain what happens when there's no C3. Go ahead, Karen.
C3 kind of starts the whole process of the inflammation and all that stuff. So if it's missing, none of that stuff can happen.
Yeah. So if we think about the central cascade as the light switch in all its guts, C3 is like sort of the outer shell where you flick it on or flick it off. And without that, you can't turn the light switch on. Even if the rest of the mechanics are there, if you can't switch it on, it's not going to do anything. So it is the most important. Let's look at some of these other answers and see if we can figure out what would the outcome be, because obviously C3 will just completely disable the complement. It doesn't matter if you find patterns. It doesn't matter if there's antibodies. It doesn't matter if there's lectins. You can't turn it on. But what if the only lesion was in the C1 protein? C1 is now defective, but the other C's are there. What would be the consequence? Think about what would happen then. You can put that into the chat if you have a guess. If you'd rather raise your hand, that's faster, but let's put it into chat, see what we got. Yes. Yes. Great. Good. Of the three pathways, the only one that relies on C1 is the classical pathway, and that's the one that C1 binds to antibodies. That's the one where the adaptive immune response can call on the innate immune response and activate the complement. It's called fixing the complement. So without C1, that means that the adaptive immune response can no longer call on the complement to help it once it's identified a threat. But does that mean that we can't turn the light switch on at all? There's two other pathways still. Those rely on bacterial patterns. So a bacterial infection could still trigger a complement activation just by a different method. Okay, let's look at what would happen if we had no C5. So no C5 means no C5a, no C5b. What would happen? Would you still be able to turn on complement? Yeah, you would, right? Those pathways are still working. Most of the bits of the core cascade are still working, but C5 is affected, which means you can still get opsonization, right? You can still get some inflammation because of C3a, but your inflammation is definitely going to be reduced because there's no C5a, and there will be absolutely no cytolysis. So you kind of cripple it, but you could still turn it on by any method. C9, if the only affected protein is C9, then what do we lose? Correct. Good. You lose cytolysis only. You can still turn the pathway on via bacterial signatures or the antibodies. You can still do opsonization. You can still do inflammation, but cytolysis is going to just flat out fail. Great job. Okay, let's talk about interferons for a second. So there's actually three types of interferons, and it's really annoying that they have the same name because there's two subcategories, one of which doesn't do the same thing at all as the other one. What are the names of these three interferons? Let's start there. Correct. Alpha, beta, gamma. That's right. Okay, now, as I said, there's two subcategories, right? Alpha and beta do the same thing as each other, but gamma does something completely different. So let's first focus on alpha, beta. And if you have an answer to this, how alpha and beta, like how do they help? Just put your one-liner into the chat. All right, have a good one, Courtney. Yes, good job, you guys. Good. So they're specifically involved in defense against RNA-based viruses by causing neighboring cells of an infected cell to produce antiviral proteins. So they're made by an infected cell to warn the neighbors that there is a virus in the area. The infected cell is a goner. This doesn't protect the infected cell, but it is a community service to warn the rest of the tissue that there's a problem and give them a little bit of time to build their own defenses. So interferon alpha and beta are produced by virally infected cells, specifically RNA virus. And they are made in small amounts by that infected cell. They go next door to the neighbors and tell them about what's happening and give those neighbor cells a bit of time, if they have not yet been infected, to produce an antiviral protein that inhibits the replication of the RNA virus. Yes, go on without me. It's very noble, self-sacrificing. Deep in your own cellular architecture, you have some altruism built in. All right, interferon gamma does something different. Interferon gamma is actually produced by lymphocytes, and it's used to activate neutrophils and macrophages to be more phagocytic towards bacteria. Okay, so let's have a quick look at this self-sacrificial act. So you have a cell, it gets invaded by an RNA-based virus, and the viral RNA is a trigger for the cell. It sees this viral RNA, it's like, that's not my RNA, what's that doing there? Causes the cell to make transcripts and then to translate into protein, interferon alpha and beta, which are secreted proteins that go next door, bind to receptors on neighboring cells that have not yet been infected, triggers them to produce antiviral proteins, specifically proteins that prevent RNA-dependent RNA polymerase from replicating RNA, so that when this guy tries to get into this neighboring cell, that RNA genome can't be replicated, so we don't get a productive infection. Very important, but not super helpful to the infected cell. He's a goner, sad to say. All right, so let's see if we can answer some questions. Which of these, A, B, C, or D, would you say is the best definition for innate immunity? Yes, I would agree. Several defenses, as we've talked about, present in all of us from the time of birth, and they really don't change or adapt. They can degrade as we get worse at sort of maintaining our homeostasis as we age, but they don't really change. They don't learn from the environment, and they're pretty robust for most of the time we're alive. Smoking paralyzes the cilia in your upper respiratory tract. What effect will this have on the immune system? Yes, good job. So certainly major thing is the pathogens won't be removed effectively from the airway because the ciliary escalator is failing and that's its job. And this whole thing is part of the innate immune system. So it's A and C, which is letter E. Good job. Which of the following is antiviral? Let's do a little poll. So let's start with cytolysis. Antiviral, yes or no? Excellent. That one is a no, because of course, this is attacking membranes and viruses don't have plasma membranes that really matter in terms of keeping them intact. Transferrin, is this antiviral? Correct again, because transferrin is important for scavenging iron that's required in aerobic respiration in some bacteria, and viruses are not metabolically active, so they don't care about iron. Lysozyme. That's right, lysozyme is still a no because it's antibacterial, right? It's targeting a peptidoglycan, which is bacterial. Interferon alpha, is that antiviral? Yes, especially directed against RNA viruses in that self-sacrificial act that the infected cell is using to warn its neighbors. And then interferon gamma, a little bit depends because it's part of the activation of phagocytes, primarily against bacteria, but it could potentially increase their reactivity to viruses too. All right. There were several places where we saw that the adaptive and innate immune response talk to each other. What would be one example? Put into the chat one example where you saw this happening. Excellent. Okay, for sure, the classical pathway of the complement, which is activated by antibodies binding to their targets. But there's a few others. Remember that antibodies coat targets, and this increases the ability of phagocytes to find and bind them. And we talked briefly about how T helpers activate macrophages, though we will get more into that next time. But T helpers activate lots of things, including B cells, which is still part of the adaptive immune response, but also activate macrophages, which is part of the innate immune response. Excellent. Okay, so we have a few minutes left to talk about the lab report and to answer any questions. I do want to remind you that there is still a lab meeting today. It's not sort of formal and organized in the sense of me gonna, go in a big spiel about anything in particular, though there are a couple of points that I'll raise. So if you can't make it to lab meeting, maybe stick around for a minute so I can just mention a couple things. But otherwise, you should feel free to come to lab meeting if you can, because we'll have plenty of time to do a Q&A there. So if you want to sign out and come to lab meeting at 12:30, go right ahead. Just a reminder to do, whoops, I didn't put that slide in there. Do your adaptive immune response prep for Tuesday. We are coming up shortly on the Unit 3 test. But yes, go ahead and sign out if you're going to see me in lab meeting. Otherwise, let me just mention a couple of things about the lab report. I want to remind you that the tables, the results table and the flowchart are due this Sunday. They don't have to be complete, but they do have to be started and correctly formatted. And that is just worth a couple of points to make sure you're not procrastinating and you can get the most useful part of it done first. And I want to remind you about what types of references for your research section you should be looking for. Things like CDC, NIH, WHO, PubMed articles, tax published books. You can use the Burgies, and I'm going to show you that in a moment. And you want to remember that you cannot use blogs, wikis, or edu sites. That would be like a class website. And that you're going to have in-text citations in the text part of the research section, and then a complete citation that I can verify in the reference section at the end. All right, Anthony, go ahead. It looks like you have a question, and then I'm going to just show everyone where the Burgie citation is.
Okay, this might be kind of a dumb question, but I was wondering what would happen if we got to our unknown ID, but it was the wrong one?
Oh, great question. Yeah, so two things will happen. One is there's a small number of points on the lab report that's straight up. Did you get the ID right? It's 5 points out of 60.
Okay.
And then depending on exactly why that's wrong, I will sort of trace back to see where you went off track. And that can
Okay.
cause some other minor deductions, like misinterpreting a test as a minor deduction, or
All right.
that kind of stuff. Yeah.
Okay, that makes sense. Thank you.
Yeah. So you can still do relatively well, even if you got it wrong, but you will be up against some loss of points. All right. I'm just going to share. I'm going to try to screen share this. Let's see, where is it? I want to show you one thing that you, just in case you don't know and you can't see what I'm sharing right now. That didn't work. So let me try that again. There we go. That's better. In the Burgess module where you got your table, there's also for each group a subsection, an extract from the Burgess manual. The tables are pulled from those manuals as well. The manual is a multi-volume collection of all the things we know about bacteria. But there's your section, which is usually like somewhere between 50 to 150 pages. So for instance, here's the Bacillus one. You can download it as a PDF, and you can read through it for general information about Bacillus. And once you know what your species is, I have this downloaded somewhere. And again, I'm going to go through this in lab meeting. So if you're going to be in lab meeting, you don't have to stick around for this. But if you're not, let's see if I can find it. Open. Oh, no, that's not it. I'm going to have to just download it and then open it from that internal process. In everyone's group, towards the end of their section, which of this 100-page document, there is a complete list of every bacterium and a little paragraph blurb about that bacterium. So you can go and specifically search for yours. Let's see if I can get this to turn correct side up and zoom in a little bit. And you can see that we're in the Bacillus genus, and here's like each of the bacterium has its own little compartment, little blurb about it. So you can go look up your bacterium and learn some things about it. So it'll talk about some of its metabolic features, its structural features, maybe something about its spore size and things like that. All information that you can crib for your lab report. All right, let me go back to my slides and just see if there's any questions from anyone. I'm just going to stop my recording here.